right, thank you all thank you to the conveners for having me in this session, and for all of you for sticking till the very end. Pretty much the second to the last talk is pretty much the end. I'm going to switch gears and move to laboratory investigations from theoretical conversations in the previous talk, and talk about physiological mechanisms and mineral transformations of hyperthermophilic ion reduction. So this is probably a gross oversimplification, but I want to remind and reiterate that on Earth, we know that biology greatly influences geology, and it's this coevolution of life and Earth that yields organic, geochemical, and mineral clues to trace and describe some of the earliest life on our planet. Now, among the mineral clues that we turn to are iron minerals such as iron oxides that we know today have been central to some of the earliest biological metabolisms. Yet when we look at the rock record, from a mineral perspective, we don't know if we can discern a truly microbially altered mineral. So one approach to doing this would be perhaps to examine one specific micromineral process to really get at identifying its mineral signatures and the mechanisms by which they form. So I'm going to make the argument that we can explore and better understand a micromineral process such as hyperthermophilic ion reduction to do this. Now, a hyperthermophile, just to revisit this for people in this audience who may not be microbiologists, a hyperthermophile is an organism that grows optimally above 80 degrees Celsius, and an ion reducer is an organism that couples the oxidation of organics or hydrogen with the extracellular reduction of an iron oxide so as to gain energy. Now, this type of microbial metabolism occurs widely in terrestrial hot springs and deep-sea hydrothermal vents, yet our understanding of its physiology as well as mineral transformations is extremely limited. So we're actually probing and trying to understand this process a little closely using a model system, a model cranarchaeon called Pyridictium delaney, type strain SU06. Now, I want to highlight a few key characteristics about this organism before going any further. So Pyridictium delaney is an isolate that was taken from an actively venting hydrothermal chimney and that grows optimally at 90 degrees Celsius, and it's hydrogenotrophic. And when it comes to electron acceptors, it can only really use an insoluble iron oxide such as ferrohydrite and nitrate, but it is unable to use any soluble iron, such as ferric citrate, or any macroparticulate crystalline oxides, such as goethite, hematite, or maghemite. Now, when it comes to looking at its genome, we find that its genome encodes for these 17 predicted C-type cytochromes, and many of these C-type cytochromes are actually multi-heme proteins, which we know from model mesophilic bacteria are essential for extracellular electron transfer. So we started off by asking that given that this organism only grows on these insoluble iron oxides, but not on any soluble iron, does it use some of these other iron mineral phases? And to really get at this question, we decided to synthesize a range of nanophase minerals, several of these different types of iron oxides, to test for growth. And we found overall that the organism grows on a wide range of these minerals when they're presented as these nanoparticulate mineral phases, albeit to varying degrees depending on the oxide. It grew to the highest cell concentration at the fastest growth rate, producing the most Fe2 using ferrohydrite. It grew to moderate cell concentrations, growth rates, and Fe2 production using accogenate and lipidochrosite, shown in green and orange, and to the lowest cell concentrations, growth rates, and Fe2 production using goethite and hematite. Now, these results are not unsurprising given the expected crystallinity and thermodynamic stability of these phases. But what is a key finding here is that the organism can use this wide range of minerals when we think about mineral parameters such as size. Now, next we wanted to know, given that this organism can use this wide range of mineral phases, does it form different mineral end products as a result of reduction? And to really get at this question, we tried to examine the mineral end products using a range of bulk and reflectance spectroscopies, two of which I will primarily be focusing on today to show you results. This mid-infrared attenuated total reflectance and Mossbauer spectroscopies. In addition, you'll notice that many of these these plots have a range of conditions and spectra. And the reason why this is because we're looking at biogenic mineral transformations and comparing them to a suite of abiogenic or abiotic mineral transformations that may be plausible, accounting for any transformations that may result from heat, non-growing cells, or the growth medium itself. But what we find overall is that when ferrohydrite transformations are considered, that the organism primarily makes magnetite, which is seen with the two prominent absorptions here for the mineral phase, and in the attenuated total reflectance, and in the Moss power as well, we see the magnetite. But in addition to the magnetite, we also see minor amounts of ferrous phosphate and ferrous carbonate form relative to abiotic controls. With lipidochrosite, instead of magnetite, we see the formation primarily of a ferrous carbonate phase, such as siderite, in the attenuated total reflectance spectroscopy. And in the Moss power, we see that same phase, but also in addition to that phase, we see minor amounts of ferrous phosphate forming. With accoganiite, we see minor amounts of ferrous phosphate phase, as well as magnetite, minor amount of magnetite form relative to the abiotic controls. Now, when we put together the results I showed you with regards to the rates of reduction and the spectral characteristics together, we find that the minerals that form are really affected by the type of oxide that we start and we think about growing these organisms with, as well as the Fe2 flux. Now, with ferrohydrite, we find, which shows this fast rate of reduction and high Fe2 accumulation, magnetite nucleation and crystal growth is favored. Whereas, and also in addition to that, we see minor amounts of ferrous phosphate and carbonate forming. With lipidochrosite, which shows slow rates of reduction and low Fe2 accumulation, we see siderite and ferrous phosphate forming. With accoganiite, which shows fast rates of reduction but significantly lower Fe2 accumulation relative to ferrohydrite, we see that minor amounts of magnetite and ferrous phosphate vivianite can accumulate. So context here can really make a difference in thinking about the different types of mineral phases that can form. So from a physiological perspective, we wanted to further probe and understand, does this organism require cell-mineral contact for reduction? And to do this, we really performed these barrier experiments that grew this organism with a mineral that was either encased in a dialysis barrier so as to prevent any cell-mineral contact. These were compared to conditions where the mineral was available as free suspensions. And in addition to this, we also compared these two conditions to another condition where we had an empty dialysis barrier added to free suspensions of ferrohydrite. And this condition primarily served the purpose of ensuring that the barrier itself did not pose any toxicity for growth. Now, all of these conditions we examined as is, but also in the presence of an artificial electron shuttle, AQDS, an artificial iron chelator, NTA, and also cell-free spent supernatant, so as to account for any endogenously produced shuttles or chelators. So what we found from these results were that P. delaney was unable to grow without any mineral contact, and that you'll notice we're looking at number of cells over time, and that when the mineral was available as a free suspension, regardless of whether a shuttle, chelator, or cell-free spent supernatant was added to the medium, that the cells grew rapidly over time and showed no differences in the rates of growth. Whereas when the mineral was encased in a barrier, that cells rapidly died or could barely sustain any growth. Now, when we look at the Fe2 production over time for these experiments, particularly for the experiments where free suspensions of ferrohydrite were made available with the addition of a shuttle, chelator, or cell-free spent supernatant, that only Fe2 production was stimulated upon addition of the shuttle AQDS. So next we wanted to know, given that this organism has these 17 C-type cytochromes, which C-type cytochromes may be involved in iron reduction. And so to do this, we actually looked at, we extracted the proteins from ferrohydrite-grown cells and compared them to the protein extracts of nitrate-grown cells. And we stained specifically for the cytochromes using a heme stain. And here we found differential protein bands in these two conditions, which we excised, digested, and sent for mass spec, and found that an 8-heme C-type cytochrome was uniquely produced in the ferrohydrite grown cultures, and 3 C-type cytochromes, an 8-heme, monoheme, and diheme C-type cytochrome, were produced in either ferrohydrite or nitrate grown cultures, or in both cultures, rather. So what are these C-type cytochromes? And I'm primarily going to focus on these 2 highlighted here in this next slide. When we think about these C-type cytochromes in the context of the genome of this organism, we find that 11 of these 17 total C-type cytochromes in this organism are, these cytochromes are highlighted as yellow circles here, and the number is the number of hemes, heme groups the cytochromes have. And these 11 of these C-type cytochromes are part of these 4 putative operons depicted in these 4 colors here that encode these membrane reductase complexes, for which we have really no homologs in other iron-reducing genera, such as those of Pyrobaculum, Ferroglobus, or Geoglobus. And more interestingly, the 2 cytochromes that we found on the previous slide are actually the ferrohydrite condition C-type cytochrome is encoded by this gene right here, the 784, which is part of this putative operon that also contains a gene that encodes a 13-heme C-type cytochrome. And here we see that this is the ferrohydrite and nitrate culture, where we see another 8-heme C-type cytochrome that's part of this operon that has ORFs related to sulfur reductases. So it would be interesting to know which genes are differentially expressed as well, and also quantitatively determine the abundances of these proteins that are produced. And that's part of future work for us. So I want to quickly summarize what I've been able to share today with you, and that is that a wide range of nanophase iron oxides can be used for iron reduction at high temperatures. And the key to recognizing what I want to emphasize here is that the nanophase part of this is that we need to think about environmentally relevant phases, in that the nanoparticulate phases are likely more environmentally relevant. And that growth and reduction rates inversely correlate with oxide crystallinity and thermodynamic stability. This is not surprising, but that ferrohydrite, accogenate, and lipidochrosite make interesting phases to further explore. The end products of reduction depend upon oxide and Fe2 flux, and that magnetite, ferrous phosphates, and ferrous carbonates can form. The latter 2 of these, to the best of our knowledge, have not been reported for high temperature iron reducers. And so we'd be interested to see if this is a phenomenon that occurs overall. Additionally, from a physiological standpoint, cell mineral contact is required for reduction, and that potentially novel C-type cytochromes may be produced with ferrohydrite relative to nitrate. And we're looking into further using differential gene expression and quantitative proteomics experiments. And with that, I'd like to thank folks at UMass Amherst, Mount Holyoke College, as well as Bruker Optics, and also funding, without which none of this work would be possible. So thank you.
I think we have a question over here.
Yeah.
Sorry, I misunderstood something, because you studied the growth of the microorganisms on ferric minerals. Mm-hmm. And then you showed Musbauer spectra of biotic and abiotic. So what do these 2 different spectra, what do they correspond?
So the biotic spectra are taken from exponentially grown cells, the minerals that were transformed after cells grew on those minerals. Okay, and the abiotic condition is where there were no cells added to the same exact setup.
Okay, so it is the same mineral, just before the growth and after the growth?
No. It's actually heat reacted. So these are cultures grown at 90 degrees Celsius in anaerobic cultures. And so the abiotic and the Mossbauer spectra reflect heat transformations that may be occurring for the same duration that we see any sort of biogenic transformation.
Ah, yes.
Does that make sense?
Yes, a little more. Thank you.
Do we have time for maybe one quick question, if there's others? All right, thank you.
Thank you.